Welcome back, everyone, to sqadia.com. We are thrilled to have you join us once again for another informative video. Today, our focus will be on the intriguing subject of the central nervous system of an embryo. The central nervous system plays a vital role in the development and functioning of the human body, making it an incredibly fascinating topic to explore. So, let's delve deep into the intricacies of the central nervous system during the embryonic stage. In this video, we will talk about the gastrulation and neurulation in embryo. Let's have just an overview of gastrulation. The most characteristic event occurring during the third week of gestation is gastrulation. Is the process that establishes all three germ layers: ectoderm, mesoderm, and endoderm. But do you know and how these germ layers are generated? Let's understand it. This is embryonic disc with dorsal view. This is cranial end. And this is caudal end. The primitive streak first appears in the epiblast at the caudal end of the embryonic disc. It extends towards its center, and then develops the primitive groove in its dorsal surface. At the cranial end of this develops the primitive node. Now, if we cut and look into it, this is epiblast, this is hypoblast. Epiblast cells migrate across the disc, through the primitive groove, and into the space above the hypoblast. These become the embryonic mesoderm, creating the three germ layers. Once the cells have invaginated, some displace the hypoblast, creating the embryonic endoderm, and others come to lie between the epiblast and newly created endoderm to form mesoderm. Cells remaining in the epiblast then form ectoderm. This green-colored cells are called as endoderm. This brown-colored cells are called as mesoderm, while this yellow-colored cells are called as ectoderm. So, what were we discussing? Yes, we were discussing about gastrulation. Now we are going to discuss neurulation. Its name indicates that it's the process of neural tube formation. As we have discussed, this is ectoderm, this is mesoderm, and this is endoderm. So, mesoderm cells that migrate anteriorly and accumulate in the midline form the notochordal process, while this is primitive node. Now, notochordal process release growth factors that cause ectoderm proliferation. So, what you guys are going to call this process? Yes, it is called ectodermal proliferation. The ectoderm then thickens to form the neural plate. And in next step, a neural fold arises on either side of the central axis. Now, along the dorsal edges of the neural folds are the neural crest cells. Consider these curves as a magnet. These curves fuse in to create the neural tube. Can you see the formation of the tube? But are you thinking why there is formation of this tube? Well, this neural tube later becomes the spinal cord. These are events of neurulation. Now, let's study neurulation in more detail. The formation of the neural tube from a flat sheet of ectoderm is called neurulation. The initially simple tube will develop and form the brain, spinal cord, and is the source of neural crest cells and their derivatives. Now, moving forward, this is ectoderm, this is mesoderm, this is endoderm, and this is notochord. We have discussed the development of notochord. This diagram shows early neurulation. Now, in next step, Signals from the notochord start off. The processes of neurulation in the ectoderm. In the next step, ectoderm involutes. Starts to form a tube. In the next step, neural crest cells appear. In the crests of the waves of the ectoderm that are moving towards each other. In the next step, the neural tube has formed. And neural crest cells move away. Now, let's have the dorsal view of embryo. This is the dorsal view of embryo. Around day 22 to 23. Now, this is cranial neuropore. This is caudal neuropore. This is yolk sac. This is somite. Do you know what is somite? Well, somite give rise to the cells that form the vertebrae and ribs, the dermis of the dorsal skin, the skeletal muscles of the back, and the skeletal muscles of the body wall and limbs. Now, these two edges of the neural plate are coming together like a zip to form the neural tube. At this point, both cranial neuropore and caudal neuropore are open. The cranial neuropore closes on day 24, and the caudal neuropore closes on day 26. Neurulation is now complete. Now, let's have an overview of development of the central nervous system. This is dorsal view of embryo. This is cranial end of embryo. This is caudal end of embryo. These are somite. This is neural tube. These are neural crest cells now, if we move neural tube. And neural crest cells out. Now, moving into the details. From neurulation, the central nervous system continues to develop as the cranial end of the neural tube dilates and folds to form spaces that will become the brain. The remainder of the neural tube, caudal to the first four somite, will become the spinal cord. Neural crest cells have different destination. That will be discussed later. Thank you for sharing your plans for the next video. We're excited to learn more about the neural crest cells in our upcoming discussion. Neurulation, as we discussed in this video, marks an essential milestone in the development of the central nervous system of an embryo. It involves the formation of the neural tube, which eventually gives rise to the brain and spinal cord. However, there is another fascinating aspect of embryonic development that we will explore in detail in our next video, the neural crest cells.
In the race against time, there is a game changer. Welcome to Medical Learning Reimagined, a world where over 40 specially curated courses are just a tap away. Dive into the mysteries of genetics, or unravel the complexities of cell physiology. Why stick to the old ways when you can learn smart? Harness the power of digital education. Leap into the future, mastering medicine one swipe at a time. sqadia.com. Medicine modernized.